So I have two big projects going on right now. One is this project with the upside down jellyfish, Cassiopeia. And it's indigenous to southern Florida, and most people have seen them if they ever go down to the Florida Keys, for example. And they have this unusual property is that they have algal symbionts that live inside their gut cells that generate sugars with just sunlight. And so the nice thing about that is that these are the exact same algae that live in most of the corals that we have in the Caribbean, for example. And so when we're trying to understand coral reef bleaching and how the symbiosis between the algae and the host, how that is established and how it's maintained and how it breaks down, for example, during coral reef bleaching, we need an experimental model that we can actually manipulate. It's really hard to work on corals that are endangered species, and you feel horrible, trying to do experiments on them, some of the last animals on Earth or something. So having a robust experimental model for the laboratory is really an important aspect of our future. And we have developed a lot of genomic resources, so now we've sequenced their genome, we have a lot of information about what kinds of genes are expressed. And we're really trying to understand a little bit more about how these two organisms talk to each other and live happily ever after. So another one of the animals that we work on regularly in the laboratory is a little sea anemone called the starlet sea anemone, Nematostella vectensis. And it lives out in the estuaries here all the way up into Cape Cod. And it was the first cnidarian. So the cnidarians are the corals, the jellyfish, hydroids, and sea anemones. And we sequenced the genome in 2007, so that was the first cnidarian, the first animal in this entire group of animals that the genome was sequenced. And we learned quite a bit about about evolution in general. And I've worked on this animal for maybe, 20 or 25 years or something like that. And now what interests me most is trying to make this organism do things that it has never done before. So a sea anemone is basically a coral without all the hard parts, without the exoskeleton. And so what we have been trying to do then is sort of convert our little sea anemone into a coral by expressing proteins that are involved in the biomineralization process in hard corals in this animal that has never biomineralized before. So it's quite an experiment to try to figure out how to do this. And the thing that we have realized is that many of the human diseases involve misregulation of biomineralization. We're doing some preliminary work where we can express these proteins that make the human heart biomineralized into essentially rock. We can express those in our little sea anemone, and so we can make all these proteins for further study. And we've expressed proteins from sea urchins, we've expressed proteins from human teeth in our little sea anemone. So this shows you how powerful the system is. If you have a laboratory system that you can manipulate, you can start asking lots of different kinds of questions that you never thought you'd ever be able to tackle.